I'm grateful for this opportunity to speak today. I'm grateful for the opportunity to come to the conference. Lots of times people ask me, what do I do for a living? And jokingly I tell them I move small volumes of liquid from one tube to the next. And really that's what it feels like lots of times when we're doing this assay development. We're moving small volumes of liquid from one tube to the next. And coming to these type of conferences are great because we get to see, as somebody who's on the bench a lot, and we get to see the applications of these things. We get to hear these awesome ideas and these awesome therapies that are coming out from these small volumes of liquid that we move from one tube to the next. So I'm really grateful that you guys are here to share those big picture items with us. So what I want to talk about today is, at Quansys, how do we go about developing our assays? We often have people, such as yourselves, come to us with these fantastic ideas and say, hey, we need to develop an assay for this. Can you do it for us? And we always say, maybe. No, we always say yes. But oftentimes there's a little asterisk there. And so I thought what we could do is we'd walk through that process. And I have a case study that I'll show on how we went through this process with one of our collaborators that came to us. So what we do at Quansys is we take our assay development process and we break it up into 4 unique phases. And those of you that have been on phone calls with me, you've heard this spiel, and so I apologize, but you're getting it again. So our first phase that we go through is our proof of concept. And we're going to take these and we're going to break each one of these up into, each of these phases up in a little more detail. But really, our proof of concept, we're trying to see, can we make lights? Our technology, we're all about chemiluminescent reaction. We want to make lights. So in phase 1, we want to get the reagents in. We want to see if we can make lights. And if we make lights and if they are of sufficient quality for us, then we say, hey, we have a pretty good idea and we think it's worth pursuing this. Let's move into phase 2. And phase 2 is really where the majority of the assay development will take place. And in phase 2 is where we are looking at diluents, at printing conditions, at assay conditions, to really put the screws to this assay to see if it's going to meet the specifications that are required for this assay to be effective, there we go, in your application. Finally, in phase 3, we have a developed assay. We want to characterize that assay. We want to verify that assay is going to meet specifications. So we will go through a production run. It's in the R&D group still, but it is with a controlled production protocol, and we look to see that it's meeting specifications. And finally, our final phase, phase 4, we move this process out to our production department, and it's our pilot production run. We establish QC release requirements, and then the kit's developed and we consider it complete. So let's walk through this process. Phase 1. We are mad scientists in phase 1. We start off with this opening meeting, and in this opening meeting, we get the requirements from the customer. I would like to build an assay for X, Y, and Z. Can you build an assay for X, Y, and Z? And we say, let's see. And for X, Y, and Z, what are your requirements? So we will go in, we'll create our design history file. This is part of our regulatory environment that we work in at Quansys, to record all these design inputs, all the outputs, and any changes that may take place. This happens in phase 1. Then we go out and we source our reagents. So oftentimes we'll have customers come to us. They have antibodies already developed, which are fantastic. We'll take their developed antibodies, we'll put them on our platform, optimize those, and move forward. Sometimes they come and they don't have those antibodies. We'll go out and we'll survey the market. We have lots of relationships with antibody manufacturers, and we will go out and survey the market, see if we can find an effective match pair that's going to work for us. And once we get those reagents in, then we look for functionality and specificity. Remember, in phase 1, we're looking for lights. We want to see, are these antibodies that we source going to be functional? And more importantly, are they going to be non-cross-reactive? Obviously, in a multiplex platform, we cannot tolerate cross-reactivity. Cross-reactivity is killer. You cannot get an accurate measurement, diagnosis, if you have cross-reactive reagents in your assay. And so that's one of the things that we look at early on in our assay development process is cross-reactivity in phase 1. So let's go through this this case study. So we had a customer come to us. They had 7 targets they wanted us to multiplex. These targets, they had already identified some antibodies, but we also were given license to screen some other antibodies to see if we needed to source some other antibodies to meet the requirements. These are the requirements that they had given us in phase 1. They wanted intra-assay CVs less than 15%. Cross-reactivity, of course, as I spoke earlier, had to be less than 1%. They gave us some ranges and some lower limits of detection. So how did we do this? We went through and screened their antibodies they gave us. This is from our phase 1 report. Antibodies for each marker were tested in both orientations, meaning they gave us a capture and detection from their assay. We took those in, we didn't believe them, and then one would work better as a capture and detection, so we tested them in both orientations. The detection is a capture and the capture is the detection. The orientations used previously worked well as an alternative orientation for all markers except possibly target 7. Using candidate antibody 2 as both capture and detection increased sensitivity for target 7. And we'll see that data here. So pulling out some of the data from our phase 1. So for example, this would be an output of our cross-reactivity, and this would be for target 6. And what we do is we will screen our match pair, so we have our capture antibody and our detection antibody, for each individual target that is going to be included in that assay. And what you're looking for is right here, this 100%. This is, we treat the signal seen for target 6 detection with target 6 antigen as the maximal signal, and what's reported here is the percentage of that maximal signal. That makes sense. So for example, let's say we see, for ease of calculation, we see a signal of 10,000 here, and we see a signal of, that'd be 100. Yes, 100 here. Then that would be our 0.1% cross-reactivity. So you can see for target 6 here, it looks great. Again, we had range requirements. They gave us some samples. These are serum samples they gave us. They wanted to ensure that the assays we developed were diagnostically relevant, meaning that their samples fell within the most linear portion of the curve that we may produce. Remember, this is still phase 1 development, so we have not finished our final optimization of the assay, but this is for target 1. This is the range we were seeing in phase 1, and this is where the samples fell. Target 3, again, we felt very confident we were going to be able to get a diagnostic range that was going to be acceptable to the customer. Their samples, again, were falling here in this linear portion. Target 5, again, these ones are maybe a little bit lower. And then we get to target 7. If you remember at the beginning, I quoted from our phase 1 report that the match pairs that they had given us seemed to work best, but potentially target 7 would be better. So this was using the orientation that they had recommended for us. And you can see a lot of their samples are falling right down here on the bottom end of this range. Typically, you do not want to see that. But we did see, now they didn't move up here to linear, but we were able to move a lot of these ones that would have been missed, you would have been very difficult to quantify, further up into the range, just switching the orientation of the antibodies for them. Now, for this customer, it was critical that we use their antibodies. They had already established data based on these antibodies and the epitopes they bound. They did not feel comfortable with us switching antibodies. So we were constrained with that, but we were able to meet their requirements, just changing the orientation and increase sensitivity for the assay and meet their requirements. So that's into phase 1. Then we move into phase 2. You can see we're a little more controlled as scientists. Catching the theme here? Anyways. So just like in phase 1, in phase 2 we'll have an opening meeting. And in this opening meeting, we will review the results of phase 1, and again, we review the customer's requirements for the assay. And we establish those performance specifications. Now, it's important to note that these specifications we establish are the same for phase 2 and phase 3. We want to show at the end of phase 2 that we've met the specifications. At the end of phase 3, we want to show that in a controlled production environment, we are still meeting those specifications. So the specifications are the same for phase 1 and phase 2 and 3. What kind of activities take place in phase 2? A lot. We look at printing and blocking optimization. Formalization of lyophilized calibrators or standards. Something that's happening, we're being pulled more into this. A lot of our customers want to have a unique standard for each point of their curve, meaning typically what we do is we'll make one standard and then dilute that down serially to make our standard curve. We have customers now that are wanting a unique standard for each point that we lyophilize. So that's something that we are having to undertake. We will optimize our curve, again, to make sure that we maximize that diagnostic range that is relevant to the customer. Diluent optimization is critical, and you'll see here in the results, diluent optimization plays a significant part in recovery and linearity. These things that are critical to characterizing the functionality of your assay. We look at incubation times, assay formats. We may find, and I think, my talk I give tomorrow, we'll talk a little bit about this. We may find that we need to move an assay from sandwich to a competitive in order to encompass the range that is required. And so that would happen here in phase 2. And then our reported molecule optimization. So every aspect of the assay we are optimizing. Yes?
When you do your printing, I know you have a format that's your own. Can you orient the antibody so you only immobilize it through the FC, so you maximize the antigen binding sites?
Right. Great question. No.
Okay.
There are ways, we currently do not do that, but you could think of ways of doing that. For example, you could coat the plate with an anti-FC antibody that potentially would then orient the antibodies with the FCs binding down. But again, you don't have maximal control. Typically, what in the industry, when people will do that, they will use recombinant antibodies that they then will engineer with like a cysteine, so they can use a disulfide bridge and bind that down on the plate to orient the antibody in that direction. We currently don't employ that. In the literature, from what I've seen, there really isn't a significant benefit to orienting the antibodies. There is enough functional antibody bound down in all the orientations that you don't necessarily see a significant improvement in sensitivity or specificity in you employing those. So, great question though, great question.
Very basic technical question that I've always wanted to ask ELISA manufacturers. When you lyophilize the standard, why do you put it in that type of container? And is there any concern with the product sticking to the rubber stopper that when you reconstitute different vials that you don't have the same concentration?
Right. Great question. So primarily the reason lyophilization vials, and they're not all the same, but they will use a rubber stopper, is so you can use shelf stoppering. So you can put that stopper in while the lyophilizer is still under vacuum. So what they'll have is you'll have a shelf in there, you have your vial sitting in there lyophilizing, and then you have that rubber stopper. And if you look on the sides of those rubber stoppers, they have little ridges, and they're pre-populated in there. And then when you're done, the machine goes up and rams all those stoppers into the shelf above it and pushes all those stoppers down. And you do that under vacuum, so you don't reintroduce water, humidity into the vial, and it maintains the stability of the product in the vial. So a good strategy to ensure, because it is problematic if you get material up in the lid, is reconstitute and then invert your vial and let it sit there, and then,
The volume that we're dealing with is so small.
It can be small.
100 microliters
Right.
for our particular assay, so I kind of have a real concern when we're doing that.
Right.
Is that an unwarranted concern?
It's not unwarranted. As we say all the time at Quansys, if you're going to do something and it's wrong, that's fine. Do it wrong the same every single time. Obviously it's better to do it right, but with an ELISA, be consistent in what you're doing. And that's exactly what it sounds like you're trying to do, because you want to make sure you get that little flake off the top of that thing. So I would, if I were me doing it, I would invert the vial and then pull it back down. Even with 100 microliters, you should have sufficient to be able to pull out what you need. Good question.
We have a centrifuge step that will improve things quite a bit.
If you can put the glass vial in a centrifuge.
We'll take a centrifuge.
Well, if you wanted to do that, you put it in a 50 ml conical, and then put it in a centrifuge that would accommodate a 50 ml conical.
Okay, what speed do I want
Super low. And that is technical speed. I think it's on your centrifuge, super low.
Yes, we have that one.
There you go, good. Good. When we are done with all of these optimization and the development here, then we move into characterization. And this is where we are looking at the specifications that the customer has provided us. We look at specificity, calibration curve, do we have a good backfit of our curve, sample linearity, spike and recovery, precision, all of these type of characteristics that we're very familiar with as ELISA users that are very important to us to know that the assay is performing well and meeting our specifications. The outputs for phase 2, we will give a report that has how all of these different characterizations performed and a production protocol, and then oftentimes we'll supply alpha kits to a customer at that point. Okay. So back to our case study here. These were the requirements that were given to us by our customer for the development of this assay. As you can see, we have, again, cross-reactivity. We have our precision requirements here. They want us to look at drift, edge effect. This is spike and recovery, linearity, standard curve backfit. We also do stability. Our stability protocol is a combination of accelerated and real time. So as we are obviously our accelerator will end, but those reagents that were used for the accelerated will just continue on for our real time going out 2 years? 2 years. And then, as I stated, they had already developed some ELISAs. They wanted to have 80% concordance with their previously developed ELISAs. So let's see how this did. So in phase 2, we had to go through and select diluents that maximize correlation, linearity, and recovery. So we did that by looking at 8 serum samples, and we went through and we looked at these different formulations and different blockers. We found that blockers for this one didn't have a significant impact on those performances. As long as we were able to get the background down, it didn't have significant impact. However, on the calibrator diluent, it was very, very important which formulation of the diluent we used. The states here is target 1, 2, and 5 had very little change in correlation with the diluent we used, where targets 3, 4, and 6 had significant changes based on the diluents that we use in terms of concordance and assay performance. And here's the data from that. So here's target 5. The red line was, I'm trying to remember how this worked. Candidate 1 and candidate 2, okay, sorry, this is where we wanted to look here. So this is, I believe on the x-axis is their previous data. On the y-axis was the data we provided, and here's the concordance, in terms of R squared, 0.9, if you remember our specification was 0.8, so we met. And again, you can see here in this graph, what we're showing here is our sample diluent candidate 2 and sample diluent candidate 1 did not have significant impact on the calculated value for the analyte that we were measuring. But on concordance, there was some differences. So obviously, candidate 2 was a better sample diluent than candidate 1 for target 5. Here again, target 4. Again, we don't see a lot of difference here, but interestingly enough, on concordance, it is slightly better with candidate 2. And then we start seeing some differences for target 3 here. You can see that they no longer trend with each other, always. And you can see that we get good concordance again with candidate 2, where candidate 1, that's not a very good graph. Again, we see the same thing with candidate 6. So not to bore you with that, but then we looked at spike and recovery and linearity. So here is target 1. Here's our endogenous levels that we saw in these 3 samples. We did a high spike. Here's the recovery we saw with those high spikes. And then here is the linearity once we diluted those samples down for target 1, target 2, target 7. So all of these look really, really good in terms of recovery and linearity. Then we move on to precision. Again, this is based on, we have our precision values based on concentration. So these are backfit values off the standard curve. And then over here, this is based on signal that we observed on the assay. And you can see that the specification was less than 15 and less than 20, for 15 for intra, 20 for inter, and we did well in meeting those specifications. Okay, now we're done with phase 2, moving into phase 3. And we at Quansys, we think of this as a design freeze. So in homage to our friends on the East Coast this last winter, I decided to show them a picture of, I believe this is Niagara Falls, Upper Falls, frozen. So we freeze. What does that mean? Into phase 2, the primary output for us is a production protocol. And we are done with all optimization. And what we want to do now is we want to freeze the tinkering that happens in the lab and produce the product in an R&D setting still. This isn't transferred to our production department, but in an R&D setting following that protocol that we built. And can we reproducibly meet the specifications that we demonstrated we can meet in phase 2? Again, we start with an opening meeting to review our requirements. We go through this R&D production run, we follow that protocol we built, and again, we look at all those same characteristics that we looked at in phase 2. Again, back to our case study, something that we do in phase 3 that did not happen in phase 2 in this is we looked at interference. The customer wanted to ensure that bilirubin, intralipid concentrations, and hemoglobin did not have a significant impact on the assay. And their specification is that they want to see less than 30% change in calculated concentration. And you can see for all of these, they were met. This is drift. So our drift study, we will add sample to the plate, wait 15 minutes, then add the calibrator and sample again. And what we're looking for is typically less than 15% change. Sometimes specification may be less than 10% change, meaning is our assay incubation time long enough to accommodate the loading of the sample onto the plate? Because sometimes it can take a while to move those small volumes of liquid from one tube to the next. So they wanted to, within 15 minutes, they wanted to give their technicians 15 minutes to load their plate. And so they had to have less than 15% change over those 15 minutes. And you can see that we met that specification as well. Edge effects, again, looking for a source of variability in the assay. Is it possible that the outer rim of the plate would heat up faster if samples were loaded cold, or is there evaporation taking place that would cause these things that cause the outer rings of a 96-volt plate to generate more signal than the inner rings? Again, there was no edge effect present on this. One of the requirements this customer had was very, very critical for them that we show that we can have no lot-to-lot reproducibility problems. Meaning, we make it one time, and then we make it the next time, and if the customer were to use a plate or a kit from one lot and a kit from the other lot, would they see the same values? So we produced 3 lots, A, B, and C, and we ran that through our precision testing, and here's the concordance between obviously lot A and B, lot A and C. Another way to look at concordance, they were quite pleased with our lot-to-lot reproducibility for this product. Here's some more of that. Lot-to-lot, A versus B of precision, A to C for target 5, and again for target 6. Okay, once we are done with phase 3, this is our verification. We now do tech transfer over to our production department, and it takes place a lot like this. We like to take the product and throw it over the wall, and no, I'm joking. There's a tendency to do that. Especially as an R&D scientist, you're done. I've beat this thing to death. I want this to be done. I'm going to throw it over the wall and let production take this thing down. And unfortunately, that doesn't work. So at Quansys, we work very, very, very hard to ensure that our tech transfer is more like this. It's a relay. You know, we want production to come up to speed with us, and we continue to run, and we hand off the product to production to ensure that we don't have that fumble that often takes place when you have a tech transfer. This is straight from our protocols. Joanne, you'd be proud. She's our quality manager, so she'd be proud that I'm quoting one of our protocols here. Design transfer is the process of transferring product design information, product specifications, to manufacturing for the purpose of manufacturing the product. This transfer does not occur at a single time, but takes place throughout the design process in the form of documentation, training, and possibly personnel. So we will actually take an R&D technician who oversaw the project, and they will go work side by side with the production technicians to ensure that all of that knowledge that we accumulated during development of that product gets transferred to the production team to ensure that when they now go and produce this product, it performs the same as it did in phase 2 and in phase 3 in our development process. So what production does to get up to speed is they will start building these, paperwork type things. What are the kit components? What are the label requirements? They'll create manuals, create certificates of analyses. These processes will start as we are coming to the close of phase 3. So when we transfer, they just keep going seamlessly. And of course, they work very closely with the customer to make sure that we meet all these requirements they would have for labeling. A lot of our product we make is for OEM purposes, meaning we will take the customer's labels and apply it to the vials in the product that we make. So we want to make sure that we meet those specifications that the customer is going to have for those type of things. The internal activities, this is where production is with R&D. They're making their bill of materials. The QC is making their pass-fail criteria, the release criteria for releasing this product. And when all of that is done, they go ahead and they make it, and your product is delivered to you. And that is, at that point, we consider this product to be a production-only product. We basically turn the crank, and we ship you millions and millions and millions of your product. There we go. I got a nod. I was hoping I'd get a nod.
At a substantial discount.
Yes, when you get to 2 million in the first 6 months, there is a price break, I believe. So, just to review, our phase 1, proof of concept. Phase 2 is really where the bulk of our work takes place. Phase 3, we want to verify that what we made does what we said and what we intended it to do. And phase 4 is our transfer. And again, just to emphasize, we strive at Quansys to make sure that transfer is seamless, that the knowledge that is gathered in the R&D group as we develop this gets transferred to the production group, so the product, it just improves the quality of the product that's made.